All right, here we are with chapter number 8, nucleotides and nucleic acids. We're going to jump into nucleic acids and nucleotides, and we're going to talk about their structure. We're going to go through classification. We'll even talk a little bit about technology related to them. That will be a little bit more chapter 9, but understanding the foundations is a critical part of nucleic acids. You've probably already been exposed to them. If nothing else, you've probably seen the movie Jurassic Park, where they talk about dino DNA, which is one of my favorite parts of any movie of all time. At any rate, here we go. So the functions of nucleic acids. Nucleic acids are polymers of nucleotides used for a number of different things. Storage of genetic information, and if you pay attention to any sort of well, I don't know how to characterize it, but there's actually people who are developing ways to use DNA as a means to store like data that's generated from a computer. Just because you use a binary sequence and you can relate DNA to binary, and so anyway, it's a way, another means of storing data. Non-genetic information, but also it's a great store for genetic information, for the transmission of genetic information, processing of genetic information, also known as ribozymes. We talked about a little bit of that in our foundations of this class, and for protein synthesis in the forms of transfer RNA and ribosomal RNA. Nucleotides are also used in the monomer form for cellular functions. For instance, ATP, adenosine triphosphate, it's made in the powerhouse of the cell, and then other nucleotides are enzyme cofactors and involved in signal transduction. So nicotinamide adenine dinucleotide and cyclic adenosine monophosphate. So a nucleotide is different from a nucleoside in that a nucleotide consists of a nitrogenous base, which is highlighted right here, a pentose sugar right here, and at least one phosphate molecule. So if you talk about something like ATP, adenosine triphosphate, that is an example of a nucleotide. A nucleoside, on the other hand, well, it is the nitrogenous base and the pentose sugar, but no phosphate is included. Carbon and nitrogen atoms on the nitrogenous bases are numbered in a cyclic format, and carbons on the pentose are designated N prime to alleviate any sort of confusion. So here's a pyrimidine and here's a purine. Those are the two big classifications, or the two big categories of nitrogenous bases. They have the numbering sequence as follows: 1, 2, 3, 4, 5, 6, 7, 8, 9, and 1, 2, 3, 4, 5, 6. For your pyrimidine. Now, if you look at the sugar ring, or the pentose ring, you've got 1 prime, 2 prime, 3 prime, 4 prime, and 5 prime. So the phosphate group is, well, phosphate's going to be negatively charged at a neutral pH. Now, this is typically going to be attached to the 5 prime position. Whenever you see that prime, you should be thinking, oh, that's on the ribose sugar. So the 5 prime carbon. Nucleic acids are built using the 5 prime triphosphate version of the nucleotide. So ATP, GTP, TTP, CTP. If this is a little bit of foreign terminology, we're going to get to that in just a little bit. 2 of the 3 phosphates are used for building nucleic acids, used for building nucleic acids form a leaving group, and completed nucleic acids contain 1 phosphate moiety per nucleotide. They may be attached to, these phosphate groups may be attached to other positions for specialized function. For instance, cyclic adenosine monophosphate, that's in the 5 prime position, but also in either the 1 or the 3 prime position as well. Here are some other nucleotides: adenosine 5 prime monophosphate, 2 prime monophosphate, 3 prime monophosphate, and adenosine 2,3 cyclic monophosphate. So these are all examples of AMP, adenosine monophosphate. We've got our adenine nitrogenous base, and that phosphate group is attached to either the 5 prime, the 2 prime, the 3 prime, or the 2 and 3 prime positions. Pentose forms differ in some nucleic acids and nucleotides. For instance, beta D ribofuranose is found in RNA, whereas beta 2 prime deoxy D ribofuranose is found in DNA. So deoxy is where the D in DNA comes from. Different puckered conformations of the sugar ring are also found. So here would be the puckered form of the sugar, where you have that 2 prime endo or exo form, or the 3 prime endo or exo form of that sugar. The nitrogenous bases, like I said previously, fall into two major categories, the pyrimidines and the purines. Now, all nitrogenous bases are simply derivatives of those. And these are heterochromatic nitrogen-containing molecules. They are planar or almost planar structures, and what they do that's particularly important is that they absorb UV light, ultraviolet light, within the range of 250 to 270 nanometers. Generally, people will say they absorb light at 260 nanometers. So that's a good way that you can use like the Lambert-Beer law to determine the concentration of a nucleic acid or of nitrogenous bases within a solution, because they absorb light at 260 nanometers. So for our nitrogenous bases, we've got several of them. We've got cytosine, adenine, guanine, thymine, and uracil. Okay, so there's a total of 5 of them. Cytosine, adenine, and guanine are found in both DNA and RNA. Thymine is found only in DNA, and uracil is the corresponding nitrogenous base to thymine, which is found in RNA. Now, these are all great hydrogen bond donors and acceptors. They are neutral at a pH of 7, and just a second. Okay, so they are neutral at a pH of 7. And here's your general structure for 2 of those molecules, your purines. Adenine and guanine are shown here. And I'm going to go ahead and write the numbering because the numbering is of particular importance. So they are 1, 2, 3, 4, 5, 6, 7, 8, and 9. Keeping in mind these are not your prime, but instead the prime numbers are on your sugars. So the similarities between adenine and guanine, well,
here's what I found.
Oopsies. You've got this carbonyl carbon at position number 6 versus you've got this amide at position number 2. Other than that, though, they're largely very similar to one another. So you could kind of look at them as, well, yeah, being very similar to one another. Now, in terms of what are they going to be involved in terms of where are charges and what's going to potentially be involved in any sort of hydrogen bonding. Is this going to be a good hydrogen bonder? Nope. Is this potentially going to be a good hydrogen bonder? Yeah, not bad. Is this going to be a good hydrogen bonder? Yeah, not bad. So we've got, on adenine, we've got 2 potential spots for a hydrogen bond to be formed, whereas guanine, this is pretty good, and not bad. Okay, so we've got 3 on guanine. Now, if we look at the structures of our pyrimidines, now, again, with our sequencing, our number sequencing, 2, 3, 4, 5, and 6. Well, let's look at cytosine. Is this going to be good for hydrogen bonding? Yeah, not bad. This and this, those are all good candidates for forming a hydrogen bond. Thymine, on the other hand, not bad. Not bad, not bad. And uracil is largely the same. Not bad, not bad. Okay. Now, with that to say, if we have something like adenine was good at forming 2 hydrogen bonds. Cytosine's good at forming 3 hydrogen bonds. Guanine's good at forming 3 hydrogen bonds. What we have is hydrogen bonding forming between C and G, and A and T, or A and U. We'll get into that a little bit later when we talk specifically about the hydrogen bonding, but I want you to think about what the structures of these molecules are and where those likely bonds are going to be formed. All right, well, we will proceed in the next video.